Why does retinol work? Walk through any skincare aisle and every product makes a promise. Firmer, younger, brighter, renewed. The language is confident, the packaging is beautiful, and almost none of it explains how. After enough of that noise, most people develop a reasonable skepticism about all of it. What they rarely develop is a reason to trust anything in particular. This video is about the one ingredient that earned that trust, and what it actually does to deserve it. In the previous two videos, we established what ages skin and what accelerates it. Collagen scaffolding that thins, senescent cells that break it down, UV radiation that reaches the nucleus of skin cells and breaks DNA. The damage is cellular. The mechanisms are specific, which means the question that follows is equally specific. Does anything exist that reaches those mechanisms and works in the other direction? One thing does, and it has been studied longer than most people have been alive. Retinol is a derivative of vitamin A. It belongs to a family of compounds called retinoids, and that family exists on a spectrum, from the gentlest form sold over the counter to prescription-strength retinoic acid, also called tretinoin, which is the most potent and most studied form. They are not the same molecule, but they work through the same mechanism, and that mechanism is unlike almost anything else in the category. Most skincare ingredients work on the surface of the skin or just beneath it. They hydrate, they soothe, they sit in the outer layers and do their work there. Retinoids do not work on the surface. They work inside the cell nucleus, on the DNA itself. Here is the mechanism step by step. When retinol is applied to skin, it is absorbed into skin cells and converted through a two-step process, first into retinaldehyde, then into retinoic acid. That conversion matters because retinoic acid is the active form, the molecule that can actually bind to what it needs to bind to. Inside the nucleus of skin cells sit a class of proteins called retinoic acid receptors, or RARs. These receptors are transcription factors, proteins that sit directly on DNA and control which genes are switched on and which are switched off. They are part of the architecture of gene expression itself. When retinoic acid binds to an RAR, the receptor changes shape, attaches to a specific sequence on the DNA, and begins activating the genes downstream of it. The genes it activates are not random. They include the genes that code for procollagen, the precursor to collagen, which means retinoids directly increase the instructions the cell sends out for new collagen synthesis. They include genes that regulate the rate of cell turnover, accelerating the replacement of older skin cells with newer ones. And they suppress the genes that produce matrix metalloproteinases, the enzymes we discussed in the first video, the ones that break collagen down. In one binding event, retinoic acid tells the cell to make more collagen, replace itself faster, and stop destroying what it has. This is not exfoliation. It is not surface resurfacing. It is gene regulation. The same territory covered in the longevity series when we discussed epigenetic reprogramming. The idea that the instructions sitting above DNA can be shifted, and that shifting them changes how a cell behaves without changing the DNA itself. Retinoids operate in adjacent territory, not resetting the epigenome the way partial reprogramming does, but influencing which genes the cell is actively reading at any given time. That distinction matters because it explains why retinoids produce results that surface-level ingredients cannot. Moisturizer can improve the feel and appearance of skin. It cannot tell a fibroblast to produce collagen. Retinoids can, because they reach the mechanism that controls it. It also explains the evidence base. Retinoids have been studied in dermatology since the 1960s, initially for acne. The first controlled trial specifically examining their effect on skin aging was published in 1986 by the dermatologist Albert Kligman. The results showed measurable improvements in fine lines, pigmentation, and skin texture, not through surface effects, but through the cellular changes the mechanism produces. In the 40 years since that paper, thousands of trials have replicated and extended those findings. The evidence for retinoids is not a marketing claim. It is one of the most robust bodies of research in dermatology. Then there is the irritation, the redness, flaking, and sensitivity that many people experience when they first use retinoids. Most assume this means damage. It does not. Skin that has not been exposed to retinoids is skin whose cells have been running at their default pace. When retinoids arrive and begin shifting gene expression, accelerating turnover, increasing collagen synthesis, signaling cells to behave differently, the tissue is adapting to a new level of activity. The irritation is the adaptation. It is not the skin being harmed, it is the skin adjusting to the mechanism working, which is why the standard guidance is to start slowly, allow the skin time to adapt, and increase frequency gradually. Not because retinoids are dangerous, because the mechanism is real, and the real takes adjustment. Here is the honest part. Retinoids do not undo UV damage that has already been written into the DNA. The mutations from decades of sun exposure are not reversed by gene expression changes. What retinoids address is the ongoing deterioration, the collagen loss, the slowed cell turnover, the MMP activity breaking down existing structure. They work forward, not backward. They also require consistency. The gene expression changes retinoids produce are not permanent. Stop using them and the cell returns to its previous behavior. The mechanism requires the signal to keep arriving. This is not a flaw. It is how gene regulation works, and they do not replace sun protection. The previous video established that UV causes 80% of visible facial aging and does it at the DNA level. Retinoids work at the gene expression level. These are different layers of the same system, and one does not substitute for the other. Understanding why something works changes your relationship with it, not just with the ingredient, with the category. Once you know what reaching inside a cell nucleus to change gene expression actually looks like, you develop a different kind of filter, not for what feels premium, for what has a mechanism. And in a world that sells promises by the jar, a mechanism is the only thing worth trusting.